Hi, my name is Barbara McPherson, and I'm going to do a presentation over glycolysis and gluconeogenesis. Today is May the 25th, 2022. So let's get started. Okay, the outline here shows I'm going to talk about an overview of glycolysis and gluconeogenesis pathways. I'm going to discuss what is glycolysis and what is gluconeogenesis. What are their similarities? What are their differences? I'm going to discuss aldolase and how it's involved in both pathways. I'm going to discuss the different isoforms of aldolase and their expression patterns, and how aldolase B is regulated by transcription factors. So that is my outline, so we'll get started. So the first slide here is what is glycolysis. Glycolysis is the breakdown of a 6-sugar molecule called glucose. The cells in the body use this glucose molecule to obtain the energy needed to sustain life. Glycolysis is a multi-step process that occurs in a specific order in the cytoplasm of the cell. This process can occur with oxygen, which would be aerobic, or without oxygen, which would be anaerobic. Without oxygen, it would go into, instead of going into the Krebs cycle, it would go into a fermentation cycle. The reactions involved in glycolysis yield 4 ATP, 2 pyruvate molecules, and 2 NADH molecules, and 2 water molecules from 1 molecule of glucose. However, you only get a net gain of 2 ATP molecules because you use 2 ATP molecules in the one of the phases of glycolysis. There are associated with this process are 10 sugars and 11 enzymes, all performing in a series of 10 steps. And here you can see that glucose, this chain reaction, and this just shows you all of these are the sugars right here in the big blue, and then out here to the side, these are the enzymes that will do some particular things to these sugars. So as you can see, you're starting with, in glycolysis, you're starting with glucose. And the enzyme hexokinase will turn glucose into glucose 6-phosphate. And then the enzyme phosphogluconeogenesis will convert the glucose 6-phosphate into fructose 6-phosphate, And then phosphofructose kinase will convert the fructose 6-phosphate into fructose 1,6-bisphosphate. And aldolase will lyse fructose 1,6-biphosphate into a glyceryl aldehyde 3-phosphate and dihydroacetone phosphate. So it cleaves off, it'll lyse off part of this molecule to form these 2 molecules here. And then triose phosphate emerase will quickly turn these 2 molecules into 1,3-bisphosphate glycerate. And then phosphoglycerate mutase, or kinase, glyceride phosphate dehydrogenase will turn this glyceride 3-aldehyde phosphate into 1,3-bisphosphate glycerate. And then this phosphate glycerate kinase will turn this 1,3-bisphosphate glycerate into 3-phosphate glycerate. And here you can see ATP is produced. Up here, ATP is used. You can see it takes ATP to start the process, and here ATP is used here. Here you have ATP produced. And then phosphoglucose mutase will convert this 3-phosphate glycerate into 2-phosphate glycerate. And then this enolase will convert this 2-phosphoglycerate into phosphophenols, and then pyruvate kinase will convert phosphophenolpyruvate (PEP) into pyruvate, and then ATP is formed here. Okay. Now there's 2 phases of glycolysis. The first 5 steps of glycolysis are considered the energy investment phase because these steps require the use of 2 molecules of ATP. In other words, energy is required in order for this reaction to take place. So you can see here, this is the first priming reaction here where glucose is converted to glucose 6-phosphate by the hexokinase enzyme. So you can see that that's an ATP and that's an ATP, and it's showing you here that that these are using energy. They're using ATP. That's why it's called a preparatory phase. It's using ATP, and it's the first 5 steps of from glucose to glucose 6-phosphate to fructose 6-phosphate to fructose 1,6-bisphosphate to glyceride 3-phosphate to dihydroxyacetone phosphate. So those are the first 5 steps in the preparatory phase of glycolysis. And then the second phase is called a payoff phase. And then the payoff phase, the 2 molecules of glyceraldehyde 3-phosphate produced in the preparatory phase get converted into pyruvate, which produces 4 molecules of ATP, 2 molecules of NADH, but only a 2 net molecule because it used up. So here you have oxidation and phosphorylation taking place here, and where the glyceraldehyde 3-phosphate, and this was the first ATP forming reaction here. Right here you got ATP formed, and then the second ATP forming reaction is right here where you have substrate level phosphorylation, and you have substrate level phosphorylation right here. And so there's your second ADP. So these last steps where you're going from glyceraldehyde 3-phosphate to 1,3-bisphosphate glycerate to 3-phosphoglycerate to 2-phosphoglycerate to phosphophenolpyruvate, and then to pyruvate, and it's going to form 2 molecules of pyruvate. Okay, now gluconeogenesis is a metabolic pathway that converts non-carbohydrate precursors into glucose. So it's the buildup of pyruvate into glucose. This process occurs during periods of fasting when your glucose levels in the blood are low or depleted. It could happen in fasting or exercising or something like that. It's just when your glucose levels are low, this is when the gluconeogenesis will kick in and form glucose. Gluconeogenesis occurs in both the cytosol and the mitochondria of the liver, as to where glycolysis just occurred in the cytosol. So the key purpose of gluconeogenesis is to create glucose for energy when needed by the body for just everyday functions. Now you can see from this diagram here that you'll probably see some of the same enzymes and some of the same sugars. That's because gluconeogenesis will use some of the enzymes and sugars as glycolysis, but it will carry out 4 bypass steps and using different enzymes. And that's what this diagram shows, steps of the bypass steps, but it doesn't show all of the enzymes and all the sugars involved. It just shows the 4 energy irreversible steps that glycolysis cannot go through. So you'll see that here at the top, pyruvate, you have 2 pyruvate acid pyruvate molecules. And this is going to, how this works is that pyruvate is carboxylized to create oxaloacetate, okay, by pyruvate carboxylase, okay, and that occurs in the mitochondria, and it requires ATP, and it also requires a coenzyme biotin, I'm sorry, and this is right here is considered the first bypass step, okay, and then this oxaloacetate here has to go through a few more things before it can leave the mitochondria to go into the cytosol. So the oxaloacetate is decarboxylated and rearranged, and then it creates phosphoenolpyruvate, which is PEP, which is right here. But before we do that, as I was telling you that this oxaloacetate has to do a few more things. So it has to be reduced to maltate by maltate dehydrogenase in the mitochondria, and that uses up 1 NADH, and then this uses up an ATP. But once the maltate is back in the cytosol, it will be oxidized again back into oxaloacetate, and then it can go from the mitochondria into the cytosol. So, and then that'll be the second step, where the oxaloacetate is decarboxylated, and then rearranged to create the phosphoenolpyruvate, which is PEP, right here. And that's done by this enzyme right here, PEP carboxygenase. This occurs in the cytosol. It requires G2P and a cofactor of magnesium ion. And this is the second step in the bypass process. Okay. And then this, it doesn't really show it on here, but PEP is hydrated to form 2-phosphoglycerate by the enzyme enolase. And then the second, the 2-phosphoglycerate converts to 3-phosphoglycerate by the enzyme phosphoglycerate mutase. That's not shown on here either. The 3-phosphoglycerate is then phosphorylated by phosphoglycerate kinase to form 1,3-bisphosphoglycerate, and that requires ATP as well, and that's not showed on here either. Then 1,3-bisphosphate phosphoglycerate is reduced to glycerolaldehyde 3 by glycerolaldehyde 3-phosphate dehydrogenase, and then a NADH is donated. Then the glycerolaldehyde 3-phosphate is isomerized to create dihydrogen oxyacetone phosphate, and that's done by the enzyme triose phosphate emerase. And then the glyceraldehyde 3-phosphate and the dihydrogen oxyacetone phosphate are combined to form fructose 1,6-bisphosphate by aldolase. Now that is right here. It just doesn't show you the aldolase or the aldolase enzyme. The fructose 1,6-biphosphate is phosphorylated to form fructose 6-phosphate by the enzyme fructose 1,6-bisphosphate. That's right here. That's the third step of the bypass. And then fructose 6-phosphate gets converted into glucose 6-phosphate by phosphohexose emerase. And then glucose 6-phosphate is dephosphorylated by glucose 6-phosphatase to form glucose, which is right down here. That is the 4th bistep part right here. And then the newly formed glucose can enter the bloodstream and do what it needs to do to help the cells. Okay, so basically then this slide just showed you the 4 pathways that the bypass pathways that gluconeogenesis takes, that they can't do the same as glycolysis. So to show you how, when you look at this diagram, you can see how gluconeogenesis and glycolysis look similar. This is where you can really see that they use some of the same sugars, some of the same enzymes. But when you look at it, it looks like gluconeogenesis is just the reverse of glycolysis. And in some areas that is true, as you can see from the diagram, that both of the pathways share, again, some of the same steps, but in reverse. Where glycolysis up here starts with glucose and ends with 2-pyruvate, gluconeogenesis is going to start with pyruvate and end with glucose. That's one difference. Another difference is of course, the enzymes that are used in the bypass steps that gluconeogenesis goes through. Basically what it is, gluconeogenesis, it bypasses the energy irreversible steps that occur in glycolysis. That's bypass is needed because of the large amount of free energy change in those steps. If you didn't have those, if it just went through there and did a cycle from pyruvate to pyruvate to glucose, then you would just be producing ATP and heat. Basically, we would just burn up. Gluconeogenesis uses these different enzymes to run these bypass reactions, which are considered exergonic. And so here you can see, if you look, the arrows in the red is glycolysis, the arrows in the blue is gluconeogenesis. And if you're starting from glucose again, you can see that the hexokinase will convert glucose again into the glucose 6-phosphate. And then the phosphohexoase emerase will convert glucose 6-phosphate into fructose 6-phosphate. And then phosphofructose kinase 1 will convert fructose 6-phosphate into fructose 1,6-bisphosphate. And this aldolase here again will lyse off this fructose 1,6 biphosphate, splitting this into these two molecules, the dihydrogen acetone phosphate and the dihydrogen acetone. Well, that both says dihydrogen phosphate. I don't know why that says that, but they're not the same. And then it'll just, the triose phosphate emerase will then convert these two quickly into this glyceryl aldehyde 3-phosphate. And then this glyceraldehyde phosphate dehydrogenase will convert this molecule here into the 1,2-bis phosphoglycerate molecule, so on and so forth, all the way down. And then I don't know if I can move I can't move that, but anyway, the pyruvate here starts from the bottom and goes up, and you can see where they're see these two enzymes are used in Lowe's. These two enzymes are the same here. These two enzymes are the same. This one is different down here. This one is different down here. Aldolase is the same. Aldolase is the same. These two are different here. These two are the same, and these two are different. So there are some differences with these two, and there are some similarities. Now, the enzyme aldolase is used in both pathways, but in glycolysis, aldolase is used to lyse fructose 1,6-bisphosphate into dihydroacetone phosphate and glycerol-aldehyde 3-phosphate. But in gluconeogenesis, aldolase is used to combine those two molecules, dihydroacetone phosphate and glycerol-aldehyde 3-phosphate, to form 1,6-bisphosphate. So basically what has happened with this aldolase is that the same enzyme will split apart and combine the same sugars just in different directions. So glycolysis is more of a cleavage of the two molecules, and gluconeogenesis is more of a condensation of the two molecules. It's using like a condensation part to put it back together to combine these two molecules to produce the glucose. These two molecules right here, I'm sorry, to produce this fructose 1,6-biphosphate. Now, the isoforms of aldolase, I really could not find a picture for that, an image for that. I searched and just could not find a good that made any sense. But there are 3 isoforms of aldolase. Aldolase A is expressed in developing embryos, adult muscles, and some cancer cells. Aldo B is expressed mainly in the liver. Aldo C is expressed in the brain, smooth muscle, and neural tissue. And like I said again, I couldn't find an image that would fit this, that would make sense. So we'll just go to the next one. And this image here is discussing the regulation of transcription factors, and I struggled to find any information that made sense to me about that. But from this image here, I can tell you that the HNF 3 alpha and there's a HNF 3 alpha and a HNF 3 beta that will compete for the same binding site, while the HNF 3 beta is considered an activator and the HNF 3 alpha is a repressor. The HNF 1 alpha and HNF 4 down here, these are transcription factors that regulate the production of liver proteins. And that's really all I could find that made any kind of sense to me about that. That's my presentation. There's my references, and that's the end of my presentation, and I thank you very much, and have a good day.